Our next presenter is Joshua McClure. He's going to present Maxwell Biosciences.
I am Joshua McClure, CEO with over 20 years of experience, and I'm excited to tell you about Maxwell Biosciences and our CLAROMER biomimetic small molecules. Our patented CLAROMER compounds are poised to revolutionize the pharmaceutical industry as a one drug for many bugs solution that could replace current global use of antibiotics, antivirals, and antifungals. As doctors, you know that antibiotic resistance accounts for hundreds of thousands of deaths each year and is a major global health threat. Last year, over $120 billion of drugs were used to fight infectious diseases. Antibiotic pollution and the growing presence of antibiotics in waterways and soils are driving further mutations and the development of further drug resistance. The world needs a new solution. I'm happy to share that Maxwell Biosciences has developed the CLAROMER platform solution, a novel class of biostable small molecules that can mimic and improve upon any peptide in the human body. We are starting with mimicry of human cathelicidin antimicrobial peptide, since cathelicidin peptides are like the Swiss Army knife of the immune system, used to fight all pathogens. Otherwise we would die. Our immune systems are fighting them all the time. So using nature as our guide, CLAROMERs, which are not antibiotics, have been developed as a one drug for many bugs solution with enhanced potency, safety, and stability to address the most serious untreatable infectious disease threats from viral, bacterial, and fungal pathogens, including challenging biofilm formations. I don't have time to share all of our data in this short 10-minute presentation, but you should know that Maxwell Science is backed by over 250 peer-reviewed publications, with over $40 million in grants awarded mostly to independent investigators from all over the world, universities and governments. Fauci's team at NIAID is a good example, confirmed our CLAROMERs as an effective one drug for many bugs solution tested against all strains of coronavirus. So it's pan-coronavirus, so SARS-1, SARS-CoV-2, MERS, etc., and all tested strains of influenza with a single compound. Other studies conducted with CLAROMERs show they're also effective against all enveloped viruses. We have tested so far, including HSV1 and 2, hepatitis B and C, and many others that are on this list here. So if you're wondering how this is possible, confirmed study data shows this is accomplished by targeting phosphatidylserine, which is a phospholipid that is exposed in the outer membrane of many pathogens and is not exposed in healthy human cells. The exposed phosphatidylserine is what your immune system peptides also target. So Maxwell's CLAROMERs mimic that with higher potency, completely inactivating the pathogen while avoiding healthy human cells. Don't take our word for it. The University of Texas Medical Branch used an electron microscope to confirm viral membrane disruption by our CLAROMERs as a key mechanism of action, which you can see there. And here it is zoomed in. Our images confirm that CLAROMERs are highly selective in targeting pathogen membranes the same way your human immune system does. This example shows the membrane disruption of two viral pathogens, a DNA and an RNA virus. So very disparate viruses there, but a single compound is able to destroy both of them. Other studies conducted with bacteria by Stanford University's Barron Lab further show that after CLAROMERs disrupt the pathogen membrane, they aggregate the internal organelles to freeze metabolism and bind the nucleic acids, thus inactivating the pathogen and also blocking drug resistance. Maxwell's CLAROMER solution provides a strong possibility to replace global antibiotic use and end antibiotic pollution. Thanks to their proprietary mechanisms of action, studies show that CLAROMERs are much less susceptible to the development of bacterial resistance. As you can see here, there's none after 30 passes. Compare that to gentamicin there, and you can see rapid buildup of resistance. So the positive impact of reducing antimicrobial drug resistance has great potential for humanity, and Maxwell aims to be a leader in this field. There is significant preclinical safety data supporting CLAROMERs, including the study showing the compounds are well tolerated in vivo. There's multiple studies here on this slide. When tested in animals at over 100 times the therapeutic dose, CLAROMERs still cause no toxicity. In rats, we showed over 3,000 times the therapeutic concentration is where we started to see some toxicity. This in vitro toxicity data here from air-liquid interface study of pseudostratified human lung epithelium tissue showed no significant toxicity was observed at the highest levels tested, all the way up to 512 micrograms per milliliter. These results show favorable toxicity profiles in highly sensitive middle ear neurons to model treatment of drug-resistant middle ear infections. So essentially no toxicity there. So we anticipate a low probability of off-target effects in neurons. Our maximum tolerated dose studies have also shown very high tolerance for the compounds. And our lead CLAROMER compound did not cause any release of pro-inflammatory cytokines. In addition to safety data, there's also strong preclinical efficacy data. To start, the NIH completed a gold standard hamster study last year. That's that Fauci study I mentioned earlier. And they confirmed in vivo safety and efficacy of CLAROMERs as both a prophylactic that prevents all symptoms of COVID and as a post-infection therapeutic that shortens the recovery time for COVID, potentially eliminating long COVID completely. For bacterial efficacy, the study from Texas A&M examined the safe, effective treatment of Pseudomonas aeruginosa, very difficult to treat infection in mice. Results show that the CLAROMER drug candidates are able to effectively target infections of the lung. So our CLAROMERs show high level of efficacy against both gram-negative and gram-positive bacteria, and once again, they demonstrate the ability to avoid the development of resistance. And that same compound is also effective against viruses and here against fungus. So the work of Dr. Corey at the Baylor College of Medicine, a medical doctor at the Dan L. Duncan Comprehensive Cancer Center, shows our fungal efficacy. So Dr. Corey works with immunocompromised cancer patients, and he often watches his patients slowly die, not just from cancer, but from opportunistic fungal infections in the upper respiratory tract because of their immune systems are depleted due to the radiation, chemotherapy, and then the cancer itself. So these types of immunodepressed or immunocompromised infections are especially in fungus, are very deadly because there are no direct fungicidal therapies. There is only fungistatic, which slows down the growth. After a decade of clinical research, Dr. Corey finally found success using Maxwell's CLAROMER compounds, which are the first to show both safety and efficacy in mouse models of immunosuppressed fungal infections of the upper respiratory system. So here's verified data from Dr. Corey's lab showing a significant 80% reduction in fungal units sampled from the lungs of completely immunosuppressed mice. So they did not have an immune system at all. So it's only the drug is killing off the fungus here. No other drug has been shown to do that. He also reported that CLAROMER treated mice showed no signs of toxicity, further evidence that our biomimetic compounds are able to target lung infections and are well tolerated in the lung. Remember, Maxwell is a platform biotech company producing compounds with many advantages over competitors. We're able to mimic any peptide in the human body. Some of the features are low toxicity, high potency, in both in vivo and shelf stability, novel mechanisms of action that can help avert the development of drug resistance, and relatively low cost of goods with scalable manufacturing. So Maxwell has very strong IP. We own our patents outright and have a growing patent portfolio. And the Maxwell team has a deep bench of experience in pharma drug discovery and development. Good example is Ed Rudnick, our COO and head of R&D, with over 30 years of experience in drug discovery and commercialization. Ed was the US head of R&D for Shire Pharmaceuticals, and he has brought some of his former Shire colleagues to Maxwell as well. Ed has worked on over 200 drug development projects and is responsible for bringing high impact drugs, big blockbuster drugs like Adderall XR, Carbatrol, Equitro, and Claritin to market. In addition, Maxwell has built a world-class scientific advisory board, as you can see here. Our team is focused on serious unmet medical needs that optimize our chance for FDA prioritization and emergency use authorization. Chronic rhinosinusitis is our strategic lead program, a post-viral syndrome that is suffered due to polymicrobial opportunistic invasion of the sinuses. And so these are antibiotic resistant bacteria, fungus, and then also sometimes multiple types of enveloped viruses. So we have sampled all of the literature around this and shown that our compounds are effective against every single one of the pathogens that have been cultured out of a sinus in published literature. So once again, one drug for many bugs. Testing is going well for that lead compound for chronic rhinosinusitis, and our pre-IND meeting with the FDA is on track for this quarter. We got approved for that meeting. It's a written response only meeting, and we're targeting Q3 2023 for our IND application and Q4 of this year to start our phase 1 human trials. So going into
time check.
Thank you. Almost done. Maxwell has already achieved several world first milestones that de-risk our technology, as you can see here. As Maxwell transitions to become a clinical stage company later this year, our business model is designed to generate revenues from royalties up front, even before our IPO. Here's some key takeaways. Thank you for your time. I look forward to your questions.
So this is focused just on the respiratory system, or this is any organ system infection?
Right now, we're just targeted on a nasal spray for chronic rhinosinusitis.
Okay, but in the future, is this only applicable to respiratory illnesses or respiratory infections, or is it any, other tissue infection or just respiratory infection?
Everything, yeah, everything. So topical, systemic, intramuscular. We have tested every method of drug delivery and shown safety in mice and rats. We're starting with the nasal spray because it's first in human compound, so we don't want to start with a systemic administration. We want to lower the risks, mitigate the risks as much as possible in that first impression with the FDA.
How much funding do you need to get through that first FDA milestone?
We need about $10 million to get through the first FDA milestone.
Thanks.
I've got a question. So one thing I picked up on is my whole life I've had cold sores once in a while, and it's so embarrassing and I hate it so much. I saw on one of your slides that you're going after that. Something that could potentially be very, very investable would be to say, hey, I know this founder named Joshua, and he's going to be part of curing herpes, cold sores, right? What part would you play in that cure? How would I pitch you to a big VC saying you're the next great pharmaceutical breakthrough?
Well, the first thing the VC would ask you is what are the competitors? And there's many over-the-counter competitors for HSV1, HSV2. You don't even have to get a prescription for it. Then there's also multiple prescribed medications.
No. I want you to cure it.
Topical.
Can you cure it? Are you going to be a part of the cure?
We don't know if we would be able to cure something until we go through human trials. And so you have to evaluate the risk up front. And so what we decided to do was we showed that we can, knock out a cold sore really quickly. But there's a lot of drugs that can do that. So there's no drugs that can knock out a chronic rhinosinusitis. The only approved drugs are anti-inflammatory in nature.
Okay. Yeah, thank you very much. How many people are full-time at your company?
13.
That's awesome. Thank you.
Anybody else? Any other questions?
I had a question on the 40 million in grants. Did Maxwell, you guys close 40 million in grant funding? That was you guys winning all that money?
Well, I mean, our compounds did. It was independent investigators around the world that applied for those grants. We've applied for and won about $750,000 worth of grants. The rest has been, people in Japan and Australia and all over the place. And a lot of times they don't even tell us that they're applying for it. They're just, because of the number of articles we published, they said, wow, that's really cool. I'm going to apply for a grant. And then reached out to us and said, I got $5 million. You want me to test your compounds? And so we send them the compounds.
Awesome. Thank you.
Yeah, I just have sort of follow-up question. I mean, it seems almost too good to be true, right? Like this is
We
the
get that
magic
all the
bullet for
time.
any infection. You're never going to need antibiotics again. And this is, I mean, I'm an orthopedic surgeon. I'm not necessarily in this field, but it seems like, wow, we should really be hearing more about this. And what am I missing? I know it's very early on, but what is sort of the thing that could kind of deflate this balloon before it really gets that inflated? Because it seems almost too good to be true.
Yeah, so there's a few things that can deflate a biotech, right? So lack of production capability for some kind, like there's some sort of rare molecule or rare element that's part of it, or for instance, like in the world situation we're in right now, something needs to come from China, and you can't get stuff from China very well right now. Something like that having to do with logistics. The other thing is, does it work? So we've shown that it works really well, but it might not work in humans. But this is a biomimetic of a human peptide, and it's working in rats and mice and Syrian hamsters right now. So we think it's pretty higher probability to work in humans because it's a mimic of a human peptide. It is a small molecule though, so it is not a peptide. And then safety, right? So I would say probably, for a first in human compound, I'd say probably safety is the highest risk because nobody knows, what it's going to do in a human. So that's why we went with the nasal spray application. So that's a topical application. It's not, we also picked a compound that has almost no bioavailability. So even going through the nose, which is, you can have a nosebleed, right? So that gets into the bloodstream pretty easily. This compound that we're starting with, MXB 22510, doesn't go into the bloodstream at all. It avoids the bloodstream. It seeks the epithelial mucosa tissues and actually gets in there and sticks into the tissues of the epithelial mucosa of the sinuses. And that's what we want it to do, to kill off the infections in those tissues.
And it's not specific to virus, fungal, bacterial, the type of bacteria. It's the same compound for every type of infection.
It's a utility infilter. That's what our head of R&D likes to say, right? It's like that this player can sub in for any of the players, right? It knocks out all enveloped viruses that we've tested, all fungus, yeast, mold, disrupts biofilms, prevents biofilms, and is highly effective even against antibiotic resistant bacteria. It's not an antibiotic, so it's mimicking the immune system. It's not the best at viruses, not the best at bacteria, not the best at funguses, but it's generally really good. So it's a broad spectrum, and then also doesn't have bioavailability. So that's why we selected it.
Could it be used for wound? Could it be used for wound care?
Yes, that's one of the areas that we're working with BARDA and DARPA, ARPA-H, and CARB-X, and wound care is one of the things that they would like us to apply for. We only have so much money though, so we haven't done a lot of studies on wound care per se. Obviously, if we got it approved as a nasal spray, you could spray the nasal spray on your skinned knee or a septic wound or something like that if you didn't have anything else. But we would like to do some specific studies on wound care to show acceleration, controlled studies.
It'll be good to see how much money we are spending on wound care versus sinusitis, and that's your money right there. Right?
Yeah, sinusitis is by far the larger. Sinusitis is suffered by about 12% of the global population, and it seems to be about uniform. Even first world versus developing world, it's about the same. So it's massive. It's somewhere around 20 million patients in the United States. And a lot of these patients have diabetes, comorbid with long COVID and many other different types of immunosuppressed patients, because otherwise the fungus that's in there would be killed off by the immune system, right? So it's generally people that have a weakened immune system, which is perfect for us because we kind of supplement the immune system. It's almost like a synthetic immune system.
Susie, you want to ask something or comment?
Yeah, I had the same question as Yuli. Like, have you had any interest from any pharma companies? Why have you not partnered with a pharma company, just out of curiosity?
It takes sometimes years from the first meeting with the pharma company all the way through to like they actually write you a check, because every time you meet with them, they say, this is great. You checked off the 3 studies we want you to, everything with flying colors. And some new
Now go do
vice
more.
president comes in and says, we need you to do this in dogs or something. And so that's what's been going
Yeah,
on
makes sense.
for us. So we're in
Makes
talks
sense.
with many pharmaceutical companies right now. And we've knocked the ball out of the park over and over again in those talks. We just haven't signed a deal yet. We have verbal commitments, but we don't, that doesn't really mean anything.
Great. So thank you, Joshua, Tony. I see you there. Thank you for coming and presenting this fantastic work. Great
Oh,
presentation.
Tony didn't get to say anything. He's our chief medical officer. This is an MD thing. Somebody ask him a question. Come on, he showed up for the thing. Ask him some kind of doctor question.
Josh, you've done a great job. So
Yeah.
You did
May
excellent.
may I say one final thing? So Joshua, you've done 2 things. One is you've given me this like stabbing pain of fear of missing out, which is good. But on the other hand, most investors are like golden retrievers, and you need to be able to figure out how to give them a simple 1, 2, 3 reason why their investment, could turn into money. Maybe we could take it offline, because I'd love to see how we could get normal investors out in the world to invest in you.
Well, we're not actually pitching for money. So that's going to make your FOMO even, go to 11. So I can't say why we're not pitching for money, but we're not pitching for money.
Okay, well, thank you very much. This was amazing. Thank you.